It is indeed a great day to explore the world of science. I am Jovielene Peligro, the leader of Group 3 from BSN 11, and today we will be expanding our knowledge on protein analysis with my fellow members Ina Castro, Cindy Dullesco, Jullar, and Althea Yson. So, let's start. I shall first give you all a brief idea on our discussion for today. So, for the abstract, proteins are an essential component of our life, and each protein contains 20 different types of amino acids with unique structures and quantities. In the experiment that the class have performed, the presence of protein was determined through the colors that appeared when oxidizing agents were added to it, leading the students to the conclusion that ninhydrin and xanthoprotic are strong oxidizing agents in the experiment conducted. So, this experiment has expanded the knowledge of students regarding what samples are rich in protein and how to identify them, which is essential in the education of nursing students. So, for the objectives, and of course, to further introduce you to our topic, here's Miss Cindy Dullesco.
Thank you, Ms. Peligro. So for our objectives, the experiment on protein analysis is intended to distinguish the existence of protein through the use of protein tests such as the burette, 9-hydrin, xanthoprotic, and Hopkins by analyzing the colors that appear when a reagent is added. It also aimed to determine the strong oxidizing agent through the data collected. Next slide, please. So let's dive deeper. What is protein? Well, protein serves as a biological catalyst, or what we call enzymes. In layman's terms, biological catalysts are the ones that enable chemical reactions to occur without changing the equilibrium. This is why proteins are vital parts of our lives. Additionally, each protein or amino acids differ in their molecular structures, nutritional qualities, and physical characteristics and can be classified as non-essential and essential amino acids. And in this experiment, we managed to figure out how we can distinguish protein in substances as it usually shows a violet color when a reagent is added. However, since there are different structures of amino acids, some color may appear also in the process. So, there were 4 tests conducted to determine the presence of protein in the samples. And these tests are namely, first, the burette test, 9-hydrin, xanthoprotic, and Hopkins test. For now, let us find out how this experiment was carried out by the class. Ms. Yson, please proceed.
Thank you, Ms. Cindy. So, under the methodology, I shall discuss the step-by-step process and how each test was conducted. The burette, ninhydrin, xanthoprotic, and Hopkins Gold tests were used to identify if there is a presence of protein and amine or amino acids in a substance. So allow me to elaborate on the process of each test so that we can obtain a more detailed information about the method that was carried out. So, the liquid samples used in the burette test to determine whether the protein was present were distilled water, milk, albumin, and salt solution. 10 drops of the burette reagent were added to each of these samples in a dry test tube, and for the reagent to adequately rest on the sample and allow the color changes to be seen, the class allotted 5 minutes. In the ninhydrin test naman, the samples that were tested for amines and amino acids were distilled water, proline, asparagine, glycine, and albumin. In this test, 1 ml of each sample was drawn and added to a dry test tube. Droppers were used to add 2% ninhydrin reagents to all samples before being placed in a water bath for a few minutes to observe the color that would appear. And for the xanthoprotic and Hopkins Gold test, Mr. Jullar, please continue.
Thank you, Ms. Yson. For xanthoprotic test, the samples of distilled water, tyrosine, and tryptophan phenylalanine, and albumin were tested for aromatic amino acids. 1 ml of each sample was added to the test tube, along with 1 ml of concentrated nitric acid for each sample. The samples are and the concentrated nitric acid were mixed properly and put in an inclined position before being heated. Then the experiment placed the test tubes in tap water to allow them to cool and poured 2 ml of 40% sodium hydroxide into all the test tube and observe the color present each sample. And for the Hopkins, um, the sample tested for proteins were distilled water, glycine, tyrosine, tryptophan, and albumin. In this test, 1 ml mixture of each sample was added to a clean test tube and then filtered, and 1 ml of concentrated sulfuric acid and was slowly poured into each test tube. The reagent and the sample were not mixed to enable the formation of layers inside the test tube. Then the test tube were placed in a standing position and the color visible in the interface in each test tube was documented. I'm going to pass the microphone to Miss Ina Castro.
Thank you very much, Mr. Jullar. Now let's proceed with the analysis of result. Each test that was performed was able to fulfill its duty in helping the students acknowledge the existence of proteins in each sample that was used. Now let us find out more about these tests on the next slides. Okay, so for the first test that was conducted, it was the burette test. This test purpose was to determine the presence of protein in an aqueous sample. Its main component are copper sulfate and sodium hydroxide. The burette test made it easy to distinguish protein by assigning colors to a positive and a negative result. Now, if the color turns to purple or violet, it indicates a strong presence of protein, and if it turns blue, it indicates the otherwise, which means that there is no presence of protein in the sample. So let us check the results that we got from the burette test. Okay, so in testing the distilled water, the blue color appeared when the burette reagent was added. This indicates that there is no existence of protein in the sample. In the milk, the color that was visible was slightly violet, indicating that there is a presence of protein in the sample. However, the existence of protein is not that strong for it to fully show the shade of the color violet. So for the salt solution, there is also no protein present as the color turned to blue when the burette agent was added, which means that there is no protein in the sample. And lastly, for the albumin, it showed the perfect shade of violet, which indicates that there is a strong presence of protein in the sample. So let us proceed with the next slide. Okay, so for this one, this test was used to determine amines or amino acids in a substance or sample. The ninhydrin test is for the amines and amino acids. During the experiment, it was observed by the students that the colors in the test may differ from the usual color we are used to, like the purple or the violet and the blue color. However, even though there are differences in the color, it is still accepted as it is still considered as a part of considering that there is a protein in the sample. So let us observe more in the next slide. There you go. So for the sample distilled water, it showed up a colorless, which means that of course there is no existence of proteins or amino acids in the sample. So the proline showed a yellowish color and the asparagine also showed light brown. And even though these are not the usual colors that we are familiar with and where we base our judgment regarding the existence of protein, these are both accepted as positive results in a different way. So the glycine also showed the most accurate representation as it showed the violet color, which immediately means that there is an existence of protein or amino acids in the sample. However, in the last sample, albumin, there was no visible color change as when the reagent was added. The albumin just turned cloudy, but still it indicates that there is a presence of amino acids in the sample. So for the next test, for the Santoprotic test and the Hopkins test, may we have Miss Jovielene Peligro?
Thank you, Ms. Castro. So, for the Santoprotic test, this test is intended to determine the aromatic amino acids like phenylalanine, tyrosine, and tryptophan that contain phenolic or indolic groups. So, this test usually shows a positive result when it deals with amino acids that have benzene rings in their structures. So in this, colorless indicates that there are no presence of amino acids, and a yellow color or even an orange color may indicate that there is an existence of amino acids. So now let us observe and see the results that we have gathered. So in the result that we have gathered, the distilled water still tested negative for amino acids under the Santoprotic test. So as you can see, its color is colorless. So the tyrosine, tryptophan, and the albumin showed yellow and orange color when the reagent was mixed with them. So the phenylalanine, on the other hand, tested negative for the presence of amino acids as it just became cloudy and remained colorless. So for this test, only the tyrosine, the tryptophan, and the albumin were positive. So for the last test, it is the Hopkins Cole test. So this test's main purpose was to detect the substances with tryptophan-containing proteins by forming a purple-colored ring at the junction of two layers. So this indicates that there is a presence of tryptophan-containing proteins in the sample used. So for the result, so as we all know now, the Hopkins test was intended to find the tryptophan-containing proteins. So it will be much easier for us to understand how distilled water, what tyrosine, glycine, and albumin all tested negative in this test. So the tryptophan, because of its components, tested positive for protein, as it also showed a purplish pigment with a little bit of yellow, making it easier to distinguish that there is a presence of protein in this sample. So in this oral report, we're also going to answer the guide questions that was given in the datasheet for the experiment tree. So for the first question, let's have Miss Cindy Dullesco.
Thank you. So for the first question, what colors indicate the presence of protein in the given sample? So among the results shown in the experiment we have performed, it is known that in the burette test, the presence of protein is detected if the sample turns violet. Having a slight violet color in the sample still indicates that there are proteins present in the sample. In the Ninhydrin test, presence of protein is detected if the sample turns violet. If the sample turns yellow, light brown, or even cloudy, it is still considered positive in the Ninhydrin test, but in a different way. In the Santoprotic test, a sample is positive or has the presence of protein if the color turns yellow or orange. But if the sample turned yellow-orange or very orange, it indicates that this sample contains a huge amount of protein. Lastly, on the Hopkins test, Cole test, presence of protein is detected if the sample turns purple. So for the second guide question, please proceed, Althea.
Okay, so for the second question, which of the given reagents are considered strong oxidizing agents? So for this, elements having high oxidation states or high electronegativity, which gain electrons during the redox reaction are frequently used to make strong oxidizing agents. Some examples of potent oxidizers include hydrogen peroxide, permanganate, and osmium tetroxide. So from what we have researched, it is shown that the ninhydrin bears the title of being a very strong oxidizing agent. So the test samples free amino acids interacts with ninhydrin, oxidizing the substance and causing the lamination. Moreover, according to the experiment's findings, centroprotic acid and ninhydrin are powerful oxidizing agents. So this is concluded because of the number of positive samples in each test, indicating that each sample tested has the presence of protein in its components. So for the third question, Mr. Jullar, please proceed.
For the last guide question, what are the roles of the proteins inside our body? Proteins are large, complex molecules that play many critical roles in the body. They do most of the work in cells and are required for the structure, functions, and regulation of the body's tissues and organs. And they serve a variety of purposes, including acting as hormones and enzymes, preserving the right fluid and acid base balance, transporting nutrients, generating antibodies, promoting wound healing and tissue regeneration, and supplying energy when carbohydrate and fat intakes are insufficient. And we're going to the next slide. Can you proceed, Miss Tina?
Thank you very much, Mr. Jullar. So the experiment 3 that was conducted about protein analysis expanded the knowledge of students not only on the importance of protein and how it can be easily distinguished in substances, but also how the components of each sample affects the reaction when a reagent is mixed with it. This experiment also highlighted the importance of strong oxidizing agents in determining proteins in substances, leading to the conclusion that both ninhydrin and xanthoprotic agents were the strongest oxidizing agent in the experiment, as both were able to show the most positive results in each sample, compared to the Burette and Hopkins test. So for the references, may I call Mr. Jullar?
For the references, these are the references that our group has utilized in order to provide sufficient and accurate information on this oral reporting. Ninhydrin test, reaction, principle, procedure, result, and interpretations. Oxidizing agent, definition, properties, example, applications. Test for protein, an overview of protein and test for protein. Albumin blood test, Burette test, definition, and examples. Burette test for protein, principles, procedure, result, and uses. Protein analysis, Hopkins-Coles test, definition, principle, procedure, result, and uses. Ninhydrin test, definition, principle, procedure, result, and uses. Xanthoprotic test, definition, principle, procedure, and result, and uses. And I'm going to pass the microphone to Peligro.
So thank you, Mr. Jullar. So I would like to extend my gratitude to my group mates for this successful report. And then I would also like to thank Mr. Jullar for the very creative PowerPoint presentation. So thank you everyone for lending us your ears in this short oral presentation. It is always a pleasure to impart new knowledge and explore the world of science with you. Again, we are Group 3. Thank you very much.